Hello everyone, I welcome you all to Biology Lessons with Kalyani. In today's class, we will be discussing about fundamental concepts about proteins. So there is a part 2 of this lecture in which we will discuss about the protein structure in detail. So you can go through the part 1 first, later you can go through the part 2. Let's dive into the topic now. What are proteins? Proteins are polypeptides containing linear chain of amino acids linked by peptide bond. So, proteins are example of biomacromolecule, or proteins is also an example of a polymer which are made up of polypeptides. So, these polypeptides are made up of many amino acids and they are linked together by peptide bond. So if you remember the experiment which we studied in the beginning of the chapter, the proteins, as their size is very large, they will be found in the retentate fraction, right? Now, next point is proteins are heteropolymer structure. Why they are called as heteropolymers? Because we know proteins are made up of 20 different types of amino acid. So as amino acid, which is the monomeric unit of protein, as we use different type of monomeric unit or different type of amino acids to make up protein, so protein is an example of heteropolymer. If we use same type of monomeric unit, we call it a homopolymer. Here we are using 20 different types of monomeric unit or amino acid, as we are using varied amino acid to make up a polypeptide or a protein. So proteins are heteropolymer structure. Is that clear? The diversity of protein, it enables us a wide range of biological roles. We'll be looking into the different biological roles shortly, okay? Let's now look into how a peptide bond is formed. Now we already discussed that amino acids in a polypeptide, they are joined together by peptide bond, right? So here I am taking the example of dipeptide. So in case of a dipeptide, two amino acids are joined together. So look, this is one amino acid, this is another amino acid. Now we should remember that amino acid are substituted methanes. So what will it have? It will have a C alpha to it. Instead of 4 hydrogen, there will be 1 hydrogen, 1 amino group, 1 carboxyl group and a varied R group, right? So this is another amino acid. What happens, like COOH, it donates the OH group and the NH2 group or the amino group of the second amino acid, it will give the H group. So there will be a loss of water molecule and C double bond ONH bond will be formed, which will be joining the two amino acids together. So this C double bond ONH or CONH bond is called as the peptide bond. So if you pay attention here, we took the example of dipeptide, and how many peptide bonds are present here? There is one peptide bond which joins the two amino acid. Always the number of peptide bonds present in a polypeptide will be number of amino acids minus 1. So if you take example of tripeptide, which will have 3 amino acids, how many peptide bonds will be present there? There will be n minus 1 or 3 minus 1, 2 peptide bonds that will be present in a tripeptide. Okay. Now there is something called as oligopeptide, which will have up to some 10 amino acids or sometimes 11 amino acids which are joined together. More than that, if more number of amino acids are joined together, we call it a polypeptide. Is that clear? So until now, we already discussed that proteins are polypeptides which are made up of amino acid as a monomeric unit. If we look into the structure of a polypeptide, it will have two ends. Now look at the left hand side. You have the amino group present. As an NH2 group is present, it is called as the N terminus, and the amino acid present in the extreme left is called as N terminus amino acid. Similarly, if you look at the right hand side, in the right hand side you will have the COOH group or the carboxyl group present, and it is called as a C terminus. The amino acid which is present in the extreme right is called as the C terminus amino acid. Let's now look into the example of some proteins and their important functions. This is table 9.5 given your textbook. The first example here is collagen. Now collagen, we all know it is an intercellular ground substance which is found in the connective tissue, in the skin, bones, ligaments, right? Now this collagen is the most abundant protein in case of animals. If you see, the most abundant protein in general is rubisco, but if you consider just animals, the most abundant protein in animals is collagen. Next example is trypsin, which is an enzyme. Then you have insulin, which is example of a hormone. Insulin is for your blood glucose regulation, right? Then we have antibodies, which help us in fighting the infectious agents or the immunity. Then we have receptors. Example of receptors like those which are helpful for the sensory reception, like detecting the smell, taste, and even for the hormones we have receptors. So all these are made up of proteins. Next we have proteins that can act as a transport, help in transport, or they are called as transport protein. Example of it is GLUT4. This is a protein which helps in transport of glucose into the cells. So the example of GLUT4 and collagen is very important here. How can we classify protein? So based on their function, we can have different type of proteins. Examples are structural proteins which help in maintaining the structure. Example collagen, you have keratins which are found in our hair and nails. Then we have enzymatic proteins which act as enzymes such as pepsin, trypsin, okay. Digestion, in the process of digestion they are helpful. Then we have transport protein. Example of this we have hemoglobin, okay. And GLUT4 we just now discussed, which helps in glucose transport. Hemoglobin is present in our blood. Then we have hormonal proteins. Hormonal proteins example can be insulin. We have growth hormone, which is also the example of hormonal proteins. Then we have some proteins which help you in contraction and relaxation. Examples are actin and myosin, which is found in our muscles. Then we have proteins which help in storage. Example is ovalbumin, which is present in the eggs. Next we have genetic proteins, which is present in your nucleoprotein. Then we have some proteins which are responsible for the defense. Example, your antibodies and your snake venoms. Then we have receptor proteins. These proteins act as a receptor. You have receptors for your hormones, you have your sensory receptors, right? So this is one way of classifying the protein. There is another way in which we can classify the proteins based upon the type of amino acids which is present in more concentration in a protein. We can have acidic protein which will have more of acidic amino acid like glutamate, okay. Glutamate, aspartate, if it is present more, then it is an example of acidic protein. Similarly, if your basic amino acids such as lysine, arginine are more, it is example of your basic proteins. If you have more of neutral amino acids, then it is a neutral protein. Another way of classifying proteins is simple proteins. Simple proteins will have only amino acids present in them. If there is some extra non-proteinaceous group associated with the protein, we call it conjugated protein. Okay, let's look into example of simple proteins. Simple proteins example can be globular proteins which are spherical in shape. Example can be albumin, globulin. Then proteins can be fiber-like in shape. Example is fibrous protein. Example can be keratin, elastin, collagen. Okay, these are all examples of simple proteins. Why? Because they just have the amino acid residues present in them. No other extra non-proteinaceous group is present. Now, if we look into the conjugated protein, it can be lipoprotein, which will have a non-proteinaceous lipid group attached, glycoprotein, which will have carbohydrate attached, mucoprotein will also have carbohydrate attached. Now, difference between the both is that if the percentage of carbohydrate is less, typically less than 4%, we call it glycoprotein. If it is more than 4%, we call it mucoprotein. Then you can have chromoprotein, which can give your color. Example can be hemoglobin. Then you can have nucleoprotein, which will have DNA or RNA attached to it. Then you can have metalloprotein, which can have an extra metal group such as zinc, which is present in case of your carbonic anhydrase enzyme, right? So that's an example of metalloprotein. There is a third category of protein which is called as derived protein. Now, derived proteins are nothing but which are denatured or degraded product of simple or conjugated protein. For example, if you take a raw egg, after you boil it, the proteins in the egg gets denatured. So that's example of your derived protein. I hope the classification of protein is clear now. Let's now look into the structure of proteins. Now, proteins are made up of amino acids, but it can exhibit 4 different levels of organization. The first level is primary structure. Primary structure is nothing but the linear sequence of amino acid. In this stage, protein just attains the specificity, but primary structure is not enough for functionality of a protein. Now, what happens that linear structure undergoes some level of folding or twisting, that is called as secondary structure of protein. In the secondary structure, proteins can be formed as helix or they can form a zigzag pattern, that is called as your beta pleated sheet. So these are the secondary structures of protein. Third level of structure is the tertiary structure. Tertiary structure is the three-dimensional structure of a protein. In this stage or in the tertiary structure stage, protein attains functionality. So proteins are functional only in the tertiary stage. Now, in case of the tertiary structure, there is a lot of molecular interaction which take place and proteins are folded in their maximum limit. Now there is a fourth level of structure which is called as quaternary structure. This type of protein structure is exhibited by proteins which have more than one polypeptide units. So if a protein is made up of just a single polypeptide unit, it will show up to tertiary structure. Now, if the protein has more than one polypeptide, then it is called as an oligomeric protein, and the oligomeric protein will have different subunits, and the way in which these subunits are arranged, that is called as the quaternary structure, right? So this is your primary structure, just the linear arrangement of amino acid, little bit twisting. This is your alpha helix. This is your beta pleated sheet. Now again, more of folding, a complete three-dimensional structure. It is called as your tertiary structure. In this stage, protein attains functionality. So all the enzymes, they have a tertiary structure, right? Next is your quaternary structure. In case of quaternary structure, a protein will have multiple subunits. So in this case, there are 1, 2, 3, 4. 4 subunits that are present. The subunits are called as the monomers or the oligomeric units, or even protomer term is used for it. The way in which these subunits interact with each other, make bonds with each other, that is explained by the quaternary structure. So look, this is the diagram which is given in the book. Primary structure, alpha helix, beta pleated sheet. This is your tertiary structure in which the proteins look like a woven ball of wool. Okay, there are different types of stabilizing bonds present here. Along with peptide bond, you can have hydrogen bond, disulfide bond, you can have ionic bond, even the van der Waals bond. So all that is present here. This is your quaternary structure in which the way in which the different subunits are like associated with each other, that is explained in your quaternary structure. So these structures will be discussing in more detail in the subsequent slide. You can just check that out. I hope you are clear with the basic structure of the proteins today. If you enjoyed the class, please hit the like button, subscribe to the channel, and if you have any queries, please feel free to comment below. Thank you everyone. Have a good day.